Hello friends, welcome to Basic Biology. Today I am talking about a very interesting topic, cell cycle. So first of all, we have to know what is cell cycle and why it is important so. Cell cycle is a controlling system of our body that regulates the cell division. Cell division is very vital for all the living organisms, starting from protozoa to mammals. In every cases, cell division is very important, very crucial for their growth, development, the cell survival. For every activities, cell division is very important. So, cell cycle is a set of macromolecular events that leads to cell division that ultimately give rise to two daughter cells with two identical chromosomes, which are likely similar with the parental chromosomes. So cell cycle will check mainly the three major activities of a cell once it complete the cell division process or once it complete the cycle. The first thing is, once the cell is planning for division, whether that division is essential or not. You all know there is a cell signaling system in our body. So, for each and every activity, body will give signal. So, here during the cell cycle process, whether any growth signal or any cell division signal is given to that cell or not, that has to be decided during the cell cycle. So, first of all is any positive signal for cell division or any growth signal is there that supports the cell division or not. That has to be decided during the cell cycle. And the second thing is, the another important thing, when cell is preparing for mitosis, so the cellular volume has to be increased. That means the chromosomal content, the nuclear material, the cytoplasmic content, the cell organelles, the proteins which are essential prior to cell division, that has to be increased during the cell cycle. So the another important thing is the chromosomal replication. Third one is the another important thing, after completion of all those activities, that is 1 and 2, it has to be assured that the proper segregation of chromosomal material into two daughter cells. That means the daughter cell has received the identical chromosomes, the correct chromosome, that has to be assured during the cell cycle. So third point is the proper segregation of chromosomes. So these three important things that will check while the cell moving through the cell cycle. Then cell cycle consists of two phases. One is called interphase and another one is called M phase, or the mitosis phase or the division phase, you can say. Interphase consists of three parts. One is G1, that is called first gap. Second, S phase, that is called synthesis phase. And the third one is called G2, or the second gap. Now this M phase consists of two parts. One is called the division of the nucleus, that is the karyokinesis. And then the division of the cytoplasmic contents, that is the cytokinesis. Now these five phases, G1, S, G2, karyokinesis and cytokinesis, together assure the proper division of a cell. Suppose a cell is planning for division. So definitely it has to enter into the S phase for chromosomal replication. But prior to replication, cell has to synthesize some important components which are essential for activating the S phase. Now these components are proteins which are responsible for the replications, that means the replication enzymes, as well as all the proteins which are initially required synthesis of the DNA molecules. So all the replication enzymes has to be synthesized prior to enter into the S phase. So that occur within G1. So G1 is a very significant phase. It has many functions. First of all, the decision has been taken, that is whether the cell require division or not. That growth signal can be verified within the G1. Second, all the replication enzymes, proteins, the cellular organelles are synthesized during the G1 phase. That means during the G1, cell is prepared to enter into the S phase. So once it has been in S phase, the S phase is responsible for DNA replication. Now, during the replication, some pre-initiation proteins are there. Say for example, in eukaryotes, ORC, origin recognition complex. So those proteins are synthesized during the S phase. So during the S phase, only the pre-initiation proteins are synthesized that actually stimulate or initiate the replication process. After the successful DNA replication process, cell is now preparing to enter into the mitosis phase. So after replication, the chromosomal volume has been increased. Chromatids are formed. But prior to enter into the M phase, cell cycle will have to assure some other activities. That means all the cell division machineries which are used during the mitotic phase are there or not. So that has to be assured during the cell cycle process, and that occur within the another important phase, that is called G2. It's a second gap. During this phase, some factors has to be checked. First is the proteins which are responsible for the kinetochore assembly. Second, proteins which are responsible removal of the nuclear membrane. That is, some proteases are synthesized during these G2 that is responsible for the removal of the nuclear envelope. So, lobes removal. And another important thing is some proteins which are responsible for the formation of the cytoskeletal structure which are used during the anaphase movement. So the proteins for cytoskeleton. So this protein has to be synthesized prior to enter into the division phase, and that has been assured during the G2 phase. So G2 phase is also significant because prior to division, all the machineries required for the division has to be clarified. All the proteins are successfully synthesized. Now cell is moving to the M phase where the cell division occurred, which consists of two parts. One is the cytokinesis and karyokinesis. Karyokinesis means the nuclear separation, the chromosomal segregation, and cytokinesis means separation of the cytoplasmic organelles and formation of two daughter cells. So in this way, cell cycle playing some important role to regulate the division process. Now another important thing is that during the cell cycle, a surveillance mechanism also function. What does it mean? To achieve the maximum accuracy and fidelity in the chromosomal replication as well as the segregation, and also to coordinate with the growth and development, cell cycle is controlled by some surveillance mechanism. That means during the cell cycle process, each step or each phase is very significant, whether it is G1 or S or G2 or M. So each phase is significant. So cell cycle has to assure that each phase completed successfully or not. So that assurance occur within the transitional zones. This surveillance mechanism is known as checkpoints. Now there are two important checkpoints. One is G1S, another is G2M. Now checkpoints inhibits each step before it assured that the earlier step has been completed successfully or not. If it is not completed successfully, then it arrests within that stage. Say for example G1 is, if all the activities of G1 has not been completed successfully, so it has to be arrested within the G1. If it is not completed in the G2 properly, then it has to be arrested in the G2. So it does not move or progress to the M phase. So cell cycle not only responsible for making the proteins synthesis, but along with that it also regulates all the activities of the cell and what is happening continuously throughout the cell. Now for the regulation of cell cycle, number of proteins are responsible, but mainly a small amount of heterodimeric protein kinase which are responsible to regulate the entire cell cycle. So these heterodimeric protein kinase consist of two parts. One is cyclin, which is present in a very low amount when cell does not require division, and another is CDK, that is cyclin dependent kinase. Now its amount is constant, it does not fluctuate whether the cell enter into the division or not, but the CDK is unable to activate any cell cycle process. So cyclin and CDK together phosphorylates hundreds of proteins as well as activates many proteins to regulate the cell cycle process. So in our later video, I will be talking about how this CDK cyclin will control the entire cell cycle mechanism and how many cyclins are there which are species specific or which are region specific to activate the cell cycle mechanism.